Yeah, okay, everyone. So, welcome to another spatial biology masterclass. I'm here today with Rachel Penny, who is a scientific officer at the Cancer Research UK Scotland Institute. And we're going to start with some questions, polls. We are testing this, so I hope it works. And that also gives some time for people to start joining. And then Rachel is going to start her presentation very soon. So we're just curious to hear a little bit about the background of our audience today. And we would like to know, what is your biggest challenge with cellular segmentation and multiplex image analysis? I'm going to give a few seconds for everyone to, vote. And in the meantime, just so you know, Rachel is going to be presenting today a novel phenotyping method to overcome this challenge with cellular segmentation. So stay tuned. Okay, I'm just going to give like 10 seconds and then we can start. Okay, so Rachel, I'm gonna hand it over to you and let's get started.
Hi everyone, let me just share my screen. Can everyone see my screen?
Yes, we can see your PowerPoint. Yeah, now we see the full slide. Yeah.
Yep, you can't see the presenter mode or anything, it's just the slide
No.
itself. Okay.
Yes, correct.
Okay, well, hi, I'm Rachel. Thanks for having me today. I work in Cancer Research UK Scotland Institute as a translational histologist in the Deep Phenotype and Advanced Technology facility in Professor John LeCane's lab. And today I'm going to be speaking about our method that we've developed, which is a global phenotyping method using the artificial intelligence module in Visiopharm to overcome the cell segmentation bottleneck. But first, just a brief overview on some of the spatial technologies that we have. Some we use Visiopharm to analyse, and some we need to try or we haven't tested out yet. So we have the 10x genomic Xenium, which we can do up to 5,000 RNA. We have the Lunar 4 Comet. Well, we don't have the Lunar 4 Comet yet. The Lunar 4 Comet yet. Sorry. We are anticipating its arrival, but we're hoping to get that quite soon. This can do more or less up to 100 proteins, maybe more, depends on how much we want to push it to its limits. We have the Akoya phenocycler fusion, and currently we've done around 43 proteins. We have the Roche Montana Discovery Ultra, which is our lower plex instrument, but it's our more high throughput. And this can do up to 8 proteins plus DAPI. And we scan all of our lower plex assays on the Akoya PhenoImager. And our newest toy in the lab is the Olympus VS200. And today I'm mostly going to be speaking about how we use Visiopharm to analyse more of our lower plex assays. So most of the tissues that we analyse are from our lattice cohort, specifically our lattice A cohort, which is around 1,025 lung adenocarcinoma patients across 23 TMA blocks, and each TMA has 3 cores per patient. So you can see here that the patients are in triplicate with the controls, so we can orientate it. And for each patient, we have sequencing data, we have all the H&Es, and we have resection samples as well. So we have a lot of information that we can use along with the Visiopharm data. So even with our lower plex assays, or the ones that we, or the images that we generate from the Ventana and the Akoya PhenoImager, and we then spectrally unmix them using the Akoya Inform with project-specific spectral libraries. And even though they are our lower plex, we generate highly data-rich images, and I'll talk you through how we analyse most of our images as we go. So we have many tissue segmentation algorithms. We've developed loads based on the tissues and the projects that we're working on. So we've got tissue segmentation algorithms for pancreas, kidney, lung, liver, you name it. We've most likely got a tissue segmentation algorithm trained to segment the tissue. And on the right hand side here, this is a core with that's been stained with our immune panel, and it's of lung adenocarcinoma. And we use Visiopharm, again, the unit, to segment all of our images into background, which you can see outlined in green, tumour, and that you can see in blue here, and red, which is stroma. And we also segment necrosis as well, but it's just not shown in this core. And we usually train with human annotations using DAPI, autofluorescence size keratin, as the input. We will usually adjust the freeze depth as we see fit, and we try and aim for a loss of below 0.1. And we'll always get, we've got 2 pathologists in our team, John LeCain, who's the academic lead. He's a trained pathologist that he will, like I said, look at our images for us, make sure that our ground truth is in fact the ground truth. And we also have Kai, who is a pathologist who is doing his clinical fellowship with us just now as well. Once we segmented our tissue into regions, we then go into nuclear segmentation, and we use the app that Visiopharm developed, and we have retrained on a lot of our lung adenocarcinoma cohort. This is due to our low DAPI staining in this tissue. And again, we'll get our pathologist just to make sure and validate that everything's okay. So you can see here that it actually works quite well. It's worked better than we've seen in QPath, etc. So yeah, we're quite happy with it. And for the nuclear for this whole cell segmentation, we, like others, segment our cells based on the expansion of the nucleus. And we do this by ROI. So if a nucleus is sitting within a tumour region of interest, we'll expand by, say, 10 to 20 pixels. And if it's sitting stroma, we'll do maybe 5 to 10, just depending on the tissue. And we are interested in cellular phenotyping for, biomarker discovery. So we have used the phenotyping module in Visiopharm to do this. It's based on the nuclear segmentation, and then we do a whole cell expansion. So what we've just seen previously here is we'll just turn the cytoplasm label just to one whole cell label. And then the phenotyping module in Visiopharm uses the probabilistic model for clustering. So for example, if you have CD3 positive cells, they will be a more numerous class as CD3 and CD8, for instance, and they would form a subclass. We find this a little bit challenging, mostly because when you have a cell that's expanded onto a different stain, for instance, it can be misclassified. And when you're training the phenotyping module, you need to weight it equally, or you can miss phenotypes again, or you can introduce more negative and unidentified. So yeah, we'll use this as well, and we'll do more kind of standard methods by taking the mean intensities of each marker. And We do this by, again, using the Phenoplex guided workflow. We think that this has actually been an asset that's been introduced to Visiopharm for us. So again, our workflow stays the same. We segment the tissue, we segment the nuclei, and then we expand on the cell. And then we manually set the threshold cutoffs ourselves, which is a feature that we really like. So we can base it on the whole cohort or per TMA, for instance. And then it generates a phenotype list, which is quite good. However, because we have done the whole cell expansion and it's not as accurate, it's just based on expansion of the nucleus, it can introduce false phenotypes. So for instance, you can see here that this cell has actually been expanded onto, well, it's a size keratin positive cell, a little bit of Ki67 in there, but it's actually been expanded onto an SMA. So this has created a false phenotype. So we do feel that these methods are really good and they're used all over the world, but sometimes we feel they can be suboptimal with phenotyping, and it's limited to the accurate or the inaccuracy of the cellular segmentation. Other labs we know use cocktails of antibodies to generate membrane markers, and that can be used for the cell boundary, but for us, especially for our more lower plex assays, this takes up precious space in our assays and our multiplexes. So we basically want a better way that we can phenotype without really accurately or as accurately as possible segmenting the cells. So we have developed a method in collaboration with David Mason from Visiopharm, and it's called PixelMap, and I might refer to it sometimes as a global method as well. We're just playing around with names now, but yeah, so this is PixelMap, or our global method, and this is our approach to tackle the ongoing challenge with accurate cellular segmentation and therefore cellular phenotyping, because if you've got more accurately segmented cells, you'll have more accurately phenotyped cells, and then you'll have more accurate spatial analysis for biomarker discovery. So a brief overview, this is a supervised AI using the UNet in Visiopharm. It does not rely on accurate cellular segmentation. The training images contain human annotations for entire cellular regions, and it's learned patterns of positivity for each cellular compartment, and that is probably key in this method. So I'll go into more detail about why we did this. So this is just our training pipeline overview. So to get our training set, we got our associate scientists in the team to extract single channel images from multiplex images. We use 4 different multiplex images in total on our lung adenoma carcinoma cohort. And then just as a little bit of housekeeping, within Visiopharm, I then separated them into different subfolders of cytoplasmic staining, membranous staining, and nuclear staining. And so then we annotated single channel images. So these single channel images that's been extracted from the multiplex images for each cellular compartment, either creating a positive label, i.e. where the stain was localized to, or where the stain was, what was positive for that specific marker. So that would be positive, and then background. So our outputs for the training would just be positive and background. And then the pathologists and the team, so John validated the labeled training set, and I think Kai did as well. So the two of them sat down, looked at our images, made sure they were happy before we started training. And then this part is also key. So we trained all the images in the slide tray at the time. I know we don't have a slide tray now, but you would just highlight them all. And they were trained simultaneously. And this is because they were extracted from the multiplex images, so they only have one channel. And then we checked the app on multiplex images that it hadn't seen before, just to make sure we were happy. So if we wanted to have a look at how well it could call a CD4 positive cell, for instance, in a multiplex, we would go into the input channel and say CD4, and then we would look at the feature heat map, and then that's how we validated it. The pathologist did the same. The pathologist had a look and made sure they were happy with it. And then the outcome was our fully trained app that contained information on regional positivity for each cellular compartment. So this is just an example of our training images. So for cytoplasmic staining, we extracted pancytokeratin from this multiplex image at the top here. We extracted phospho-EIF2 alpha from the second multiplex image, and we extracted RPS6 from the third. So this generated our cytoplasmic cellular localization for the training set. And for membranous, we've got the same multiplex image here, a different core though. We extracted CD4 channel from that. We extracted the CD8 channel from that. And we extracted CD9, SEA9, sorry, and PDL1. And for our nuclear staining, we use Ki67 and FOXP3. And I'm just going to go into a little bit more detail, more zoomed in so you can see. So where magenta is, everything was labelled as positive, not just where the stain was localised to. Like if we had, for instance, I always use this analogy, if we had a donut, the whole donut was given, even where the hole is, and not just the ring of the outside. And then we did the same thing for a membranous stain, so this is CD4 and for Ki67. And here you can just see how, just a little bit of housekeeping in Visioform, how I separated them into subfolders. So the reason for doing this is because, so this is a really bad drawing, but you can see that if we've got CD8 staining around the outside here in yellow, and then you've got the nucleus that would be in the middle. And see, we just did a nuclear segmentation, for example. The nuclear mask or label would be sitting inside this CD8 stain. And because it's not actually touching where the stain is, if we were to just take the mean intensity of that, it would be 0. Even if we use 100% of the object, it's not touching it, so it would be 0. So the reason that we gave the entire region of positivity as being positive is so that even if we ran a nuclear segmentation, because it's sitting in the middle and that's been classed as positive, then our algorithm is going to say, okay, this cell or this label belongs to a positive, or as positive for CD8, for example. So that was the rationale behind the training labels. And then all the images were trained simultaneously at the same time, and it doesn't really matter within the AI architecture what you put as your input, as long as it was index 1. And this is because, like I said, all of the images that we're training on only has one channel anyway. So it's going to use all of the information. It's going to look at what a cytoplasmic stain looks like, what a nuclear stain looks like, and what a membranous stain looks like. It doesn't really matter what the stain or what the marker is. It's just looking at the pattern, essentially. And then we trained it over 500,000 times with a good loss. We adjusted our freeze depth every 20 or 30,000 iterations as we seen fit. And yeah, so this is the output. So we then generate a feature probability heat map, which can be then extracted from multiplex images. So your feature outputs would be background or other and positive. So on the right hand side here, you can see our nice immune panel image. And within the AI architecture, within our pixel map or global methods, we have inputted our Ki67 because we wanted to see if it could correctly extract the probability heat map for Ki67. And on the left here, this is just the multiplex image with just Ki67 turned on, just so we can compare and see if it was correct. And you can see here that the Ki67 probability heat map is actually really quite accurate. It's really nice. And we did the same thing for CD68. Same thing for CD8. So you can see that it's classing not just where the stain's localized to, but the whole cell. CD4 and pancytokeratin. So the heat map shows the likelihood of that each pixel that is part of a cell that's positive for the marker that you select within the input. And then we wanted to try out we basically wanted to try and push it to its limits and see what we could get from PixelMap or the global method. And this is an image of an organelles panel that we did as a lab that we optimized. And in the kind of pink here, the light pink, you can see the cocktail of CD98 and ATP1A. And by just inputting that channel within the AI architecture, and we viewed the positive feature, you can actually see that it's got the area of positivity really, really accurate, despite not being given a cocktail marker within the training. It's just learned that this looks the same, this region of positivity looks the same as what was given in the training set, so it thinks that it's positive for that corresponding marker. And you can see here that this is actually, despite PixelMap only being trained on lung adenocarcinoma of human species, this is a mouse liver cancer tissue actually. And you can see down the left-hand side here, this is just the original channels from this section here of the multiplex with just the channel and DAPI turned on. And then on the right-hand side, this is a probability heat map for the corresponding marker. So you can see that it's actually extracted the probability heat maps for each marker really, really well, despite not being given MYC and CD45 in the training set, and despite not being given actually any mouse tissue. And the reason for this is because it's learned the pattern of positivity for the cellular compartment, so it knows. And we found this really interesting as well. So in the middle here, this is from another sixplex, but we've just got DAPI, CD4 and CD8 turned on just for like good visualization. And by inputting the CD4 channel within the architecture and we view the probability heat map for CD4, it can pick it out really, really well. And when you input the CD8 into the AI architecture, it extracts the CD8 probability heat map really well as well, despite both cells looking the exact same and there being similar morphologies. It knows the difference between the two because you've inputted the channel into the AI architecture and it's just used the regional pattern information to extract the heat map. And what we think is really impressive here as well is this is another multiplex panel that we've done just showing CD68 in pan-CK and DAPI turned on. So we've got pan-sized keratin in green and CD68 in red. And we just inputted CD68 within the AI architecture, and then we viewed the positive probability heat map for CD68. And again, it's picked it out really quite well, despite actually not using a macrophage marker within the training. Albeit it is a cytoplasmic A marker, but it's quite messy, but it's still being able to accurately extract the heat map from this image. So now PixelMap contains information of regions of positivity for each cellular compartment and for each cellular marker. And it can extract phenotypic heat maps or probability heat maps from multiplex images. We wanted to first know how we could actually use the probability heat maps for the phenotyping, and have we really mitigated the need for whole cell expansion? This was the main focus point here. Have we really mitigated the need for whole cell expansion because the entire regions will be classed as positive for cells? So we did a minimum object size test, and we did this just using the pan-sized keratin phenotype assignment. And we had 4 test groups of different nuclear sizes, and we compared this to manual assignment of sized keratin positive cells. And it was the pathologist Kai and the team that did this for us. So to begin, we did an object detection. And this, to start with, was just a nucleus-only detection. And this was just to generate, you can see in blue here, that we've got the nuclear masks that have just been detected to detect the nuclei. And then because we're actually going to be dilating or eroding each nuclear mask, we wanted to make sure that the X and Y coordinates remain the same throughout the test groups. And we did this by using the outline with annotation tool in the post-processing steps. So we did that, and then we copied each of the images into the 4 different folders corresponding to the nuclear size test groups. So we had nucleus plus 3 pixels, so we dilated the nucleus by 3 pixels. We dilated the nucleus by 8 pixels to try and generate a whole cell label, and the 1 pixel test group. So you can see here that, again, we dilated by 3 pixels, 8 pixels, and I don't know if you can see it quite clearly, but in the middle of each pink annotation, we've got 1 pixel spot, and the blue spot is a label. And then what we did was we used the global phenotyping app or pixel map with the deep learning. We went into the AI architecture, put the channel as an input channel or index 1 to cytokeratin. We then turned the app to then, a null classification. And then we went into the post-processing steps and said, change by intensity, label cell, or this blue label that we have here. Replace with a new positive label, which you can see here is in yellow. If it's 50%, so 0.5 or 50% sure that it belongs to the positive feature or the positive feature for cytokeratin, which we thought worked actually quite well, just even by looking at it by eye. And then we generated the XY coordinates of all of the nuclear annotations, so they would all stay the same, so we could then compare them. We took the object info of each of the labels, so we knew if it was a positive cell or it was just a negative cell, and we took the counts as well. So just for QC purposes, mainly for this point, we used the QC tool Invaluable, and I just filtered by the X and Y coordinates to create this t-SNE, and then filtered by the positive label, so what was positive for cytokeratin and what by the ground truth, and what pixel map deemed to be a positive label. And just looking by eye, you can see the ground truth had 121 positive cells within that specific ROI. And then you can see here, by the looks of it, that the 1 pixel classification was actually next best in line. So that was the best test group, apparently. And then you can see here that by eye, you can see that nuclear detection looked to be the second best. So all the X and Y coordinates lined up perfectly, all was well. So we got Kai, the pathologist, to do a confusion matrix just to make sure that what we're seeing is fine and we might not need to expand on the nucleus to create a cell. And you can see here that the confusion matrices from A here and to D here, comparing nuclear detection, the dilate by 3 pixels, dilate by 8 pixels, and the 1 pixel group, comparing to the ground truth. You can actually see here that the nuclear detection only, so just a label on the nucleus, was the best and was more comparable to our ground truth annotations. You can see the next best was 3 pixels, and the dilate for 8 pixels wasn't very good at all, and what I didn't actually expect, the 1 pixel was the worst. So by doing that, have we, to begin with, how do we use the probability heat maps for phenotyping? You can do it that way, like I did. So you could do the post-processing steps depending on what you put as your input within the AI. When you turn the app to a null classification, you can say change by intensity of the label cell to, as you know, Ki67 positive or CD8 positive. And depending on how many markers you want to phenotype, you would have that amount of apps. And then the next question that we had was obviously, have we really mitigated the need for whole cell expansion? And to be honest, at this point, we would say yes. We still to do the test with all the other markers within the multiplex, but by eye and looking with Kai and John just looking at images, they would agree, yes, we have mitigated the need for whole cell expansion. We can just use the nuclear label. And then next would be, the question was, what's the output? Like, what can we get from the heat maps? And So we go back to the beginning. So we segment our tissue into tumour, stroma, background, and necrosis. We then just do the nuclear segmentation, just plain and easy, plain and simple nuclear segmentation, and then the output. So we would have our global method or our pixel map, and then this would be used as a parent app, and then depending on what you have as your input channel, so for instance, we have CD4 at the moment, we would then turn the CD4 app to a no classification app, and then as the output variables, we would then say the mean intensity of the positive feature, so the probability of CD4 in each label, so in each nucleus or each cell, whatever you want to call it. And then depending on how many phenotypes you want to get out of this, you would have that corresponding to the number of apps. And then the output would then be your probabilities. So you just get your standard TSV that you can change, that you can open in Excel, and then for each phenotype, you'll then be given a probability based on if the nucleus, if PixelMap deemed this to be positive or not, and then you could set your probability cutoff. So you could set how confident you want to be. If you want to be 50% and above, 70%. We are still currently benchmarking this. So we're benchmarking it against normal mean intensity measures that we would normally do, and comparing this to the ground truth annotations of what Kai and John are going to be phenotyping. But we are currently benchmarking what is the best, or how confident we need to be for this to be accurate, essentially. So for future work, we obviously are pushing to publish. We're hoping to have this paper out in PLOS by the end of the year. And we want to publish data from our other multiplex panels and our more high-plex panels, because PixelMap was trained, or the global method was trained just on the images that was generated from the PhenoImager. So, and we've tested this on the Comet as well, the Luna 4 Comet images, and it works quite well. Probably works on about 80% of the markers. So I think there's some room for improvement in that instance anyway. So we want to try and get PixelMap to work with more high-plex assays. So for instance, phenocellular fusion. We want to refine PixelMap for fibroblast phenotyping. So because we didn't actually give PixelMap the morphology of the fibroblast, so the long elongated cells, it's not very good at identifying fibroblasts as of yet. That's not to say that it won't be able to do it. I strongly believe that we can. We also want to try and incorporate some RNA-scope or spot analysis. We know that this can be done. We just need to do it within our method. And yeah, so cross-imaging platform method, we want to try and get it to work on, for instance, phenocellular fusion, etc. So thank you so much for listening. I'd like to give a massive thanks to the Leucaine Lab, specifically Leah, Ian and Kai, and David Mason from Visiopharm and the Tom Bird Lab for allowing us to use their images, well, our images that we developed for their project. So thank you. I'd love to answer any questions if you have any, if we actually have time.
Thanks, Rachel, for this great presentation and sharing. Well, congratulations for developing PixelMap. And well, we look forward to see the method published soon, so it can inspire a lot of other researchers around to start using it. So let's start with the Q&A. So we have a first question from Shannon. Has this workflow been tested on imaging mass cytometry data or only fluorescence images?
Currently, we've only done it on fluorescence just now for protein-based. And again, we've mostly tested it on the PhenoImager, so the imaging methods that you actually extracts autofluorescence and uses it as a channel. So yeah, we've only done it on that, but that's a really interesting point that we could hopefully test in the future, or by all means you test it, but not before we publish, please.
Cool. And the next question from Shatavisha. For the probability heat map, how are the thresholds for positivity calling determined? How can this be adjusted?
So, when you get an AI or trained deep learning algorithm in Visio-Form, you can look at the features and you hover over to see if it's, if you hover over where it's white, that's like 90 to 100% positive, and if it's black, it's 0. So right now, we just use a 50% probability for the heat maps, just as standard just now. And then, like I said, we are currently benchmarking pixel map against standard methods, like just using immunotensities to phenotype and by manual assignment of the phenotypes. And we're going to try and see how harsh we can be to still generate accurate phenotypes. So putting the confidence or the probability up to 70%, 80%, and see how harsh we can be, but still not lose accuracy, essentially. So right now, our pathologist is doing that. But yeah.
Thank you. And Sylvia is saying, hi, great talk. Why do you think at the end, first pixel was the worst with the confusion matrices when it looked better before?
Okay, so when we generated the TISNI, so because I was just looking at the X and Y, so we're just looking at where they were, like where all the cells were. It's only comparing the number of positive cells. It's not comparing if it's actually right or not. So it could be 112, I can't remember what the number was. It could be 112 incorrectly classified cytokeratin positive cells. So obviously at first glance, I was like, wow, this looks great. Like 1 pixel can be the best, but turns out with the confidence matrix that Kai generated, no, the nuclear detection was the best compared to the ground truth.
Thank you. Alessandra is asking, so phenotyping ultimately results from a decision on an arbitrary probability value?
Mm-hmm. Sorry.
Yeah, so phenotyping ultimately results from a decision on an arbitrary probability value. I think that's...
So yes, essentially. So we would look at all the probabilities generated and then set the confidence or set the cutoff and say, yes, this is correct. But I suppose you would do that with generating mean intensities for whole cells anyway. You would look at your data. Yes, there would be some normalization methods there, but you're still looking at the mean intensities, and sometimes the mean intensities can't actually be quantified depending on the scanners and stuff. So, I mean, I'm not saying by any means that it's going to overtake mean intensity generation of phenotypes. We're just saying for us right now, it seems to be working pretty well and is comparable with ground truth annotation. So yeah, we do think it's working quite well.
Great. Question from Sylvia. Why do you not annotate the whole cell when training for nuclear positivity?
We don't annotate the full cell when training for nuclear positivity because we're using it with a nuclear segmentation app anyway to generate nuclear labels. So there wouldn't really be much point because the cell would then become positive anyway if it's sitting on a positive nuclear label.
Great. And question from Matthew. Thanks for the talk. How do you go about extracting single channel images from your MIF images?
Very, very good question. I only know that Ian used QPath to do that. But if you're interested, you can pop me an email and I can copy Ian in if you want. That's beyond my realms of understanding at this point because that's scripting. But yeah, Ian did it in QPath.
Cool. Thanks, Rachel. And thanks, everybody, for joining today. This was our last edition of the year. We are hoping to continue next year with more speakers. But thanks everyone for joining. Thanks to you, Rachel, for sharing the method with us, and your presentation today was great. And yeah, we are not going to share the recording yet because the method has to be published first. But as soon as it's live, we are gonna share it, and so you can watch again. Yeah, thanks
Thank you very
everybody,
much. Thank you.
and have a good end of day.
Have a good Christmas, everyone.
Oh yeah, true. Happy
Bye.
Christmas. Bye.